All right, so this lecture is going to cover signal transduction, and I'm going to specifically look at G protein coupled receptors and two different pathways that use these G coupled protein receptors. So your book mentions, so this is section 9.11 from your book. Your book also mentions there is it covers a third type of receptor for signal transduction, which would be a tyrosine kinase. And you can read that section, that'd be good for you, but I'm not going to cover that section personally in my lecture here. Okay, so an overview of signal transduction. So again, cells are living in an environment and they're constantly being bombarded from chemical signals from the outside. Especially think how your metabolism works. If you're in a fasting or a fed state, how your cells are going to change their chemistries internally based on that kind of fasting or fed state. So if you're fasting, your fat cells are going to release fats into your bloodstream so that your liver can process it to provide energy for you. And if you've just eaten a really big meal and you're in a fed state, you're going to be storing excess fat from your meal in your fat cells. So again, these are two very different types of chemistries that have to occur, and it all happens via signal transduction. So your different organs excrete different hormones, which control what the chemistry is going on inside your cells. So it's just absolutely fascinating. It's an entire field of biochemistry. You can go to conferences that only focus on signal transduction. And the pathways can get very tedious. But so I'm going to try to keep it simple for you all. And so let's talk about the basic overview of signal transduction. So there's going to be some external stimulus, and this typically is some type of a hormone or maybe a small molecule compound. So big ones, external stimuli, you can think of maybe insulin, glucagon, maybe epinephrine, that fight or flight molecule. So there's an external stimulus that's outside the cell, and it's going to bind to a membrane receptor. So this will be a transmembrane protein that will, once bound, undergo some type of conformational change, and that's what signals the transducer. So the transducer's next. The signal gets transduced, or essentially moves, from that membrane receptor to some effector enzyme, which typically is another enzyme that's bound to or within the membrane. So either maybe a peripheral membrane protein or an integral membrane protein. That effector enzyme creates a second messenger, creates many second messengers. So like one external stimulus can end up creating hundreds of second messengers, and those messengers go on and affect things within the cytoplasm. And so maybe they're turning on or turning off different enzymes, just depending upon what the external stimulus was. And that elicits some type of a cellular response, an overall cell response. So maybe in the case of a fasting state where you're having glucagon hormone in the blood, interacts with a fat cell, the glucagon interacts with the membrane receptor of a fat cell. Ultimately, the cellular response would be to export fatty acids into the bloodstream, which will eventually get taken up by your liver to oxidize for energy. Okay, so that's just the general overview. So external stimulus binds to a membrane. That usually undergoes some type of conformational change, and that signal gets transduced to an effector enzyme, typically also somehow associated with the membrane. That effector enzyme creates a secondary messenger. There could be multiple secondary messengers too, just depends on the pathway. Those messengers go and affect some type of chemistry within the nucleus or maybe the cytoplasm, and then that ultimately elicits some overall cellular response. Again, we're focusing on the G protein coupled receptors, which talks about the transducer is a G protein complex. So GPCRs, or G protein coupled receptors, that's what this image is. There's that integral membrane protein. These are the receptors, is the G protein coupled receptor. And then the G protein complex, which has 3 subunits, alpha, beta, and gamma, that's going to be your transducer. So the transducer for the next couple of things we're going to look at is always going to be this G protein. All right, so the first pathway we're going to look at for the GPCRs is the cyclic AMP. So we're going to create cyclic AMP. So this would be adenosine monophosphate. And so it's cyclic because the gamma phosphate it forms the cyclic ring within the structure. So this is the secondary messenger we're going to make that's going to create all kinds of havoc within the cell and turn things on and off and elicit some big response from the cell. So let's see how this cyclic AMP is made. All right, so this is a figure straight from your book, so just follow along. So you can have, so here we'll get our, I guess, let's situate ourselves. So here, this R stands for the receptor. This could be for maybe a stimulatory hormone, and this could be some type of maybe inhibitory hormone that uses this pathway. But so you're going to have an external receptor, and so the ligand, the hormone, will bind to the receptor, and that's going to create some type of conformational change, which is going to kick off this G protein complex. So here is the G protein. This is the transducer. This is going to move our signal to the next effector enzyme. Once the ligand binds the receptor, the alpha subunit of the G complex releases GDP and binds GTP and is therefore activated. So that is an active G protein, the alpha subunit. So when it has GTP bound, it is activated and it is released from the receptor and travels until it reaches its effector enzyme. And when you're going to make cyclic AMP as your secondary messenger, adenylyl cyclase is your effector enzyme. So again, ligand binds receptor, conformational change happens. It releases the G protein complex, that the transducer, from the receptor. GDP is released and GTP binds, which activates that transducer subunit. Transducer subunit moves along the membrane until it hits adenylyl cyclase. This is the effector enzyme that's associated with the membrane. And that takes ATP within the cell and makes cyclic AMP. So our secondary messenger is that cyclic AMP molecule, and I'm going to walk you through the mechanism of how that cyclic AMP molecule is made from ATP in a couple of slides, but we'll get there. Okay, so now we have our secondary messenger, cyclic AMP. So cyclic AMP, it goes to bind, this is our cytoplasmic effector, protein kinase A. So protein kinase A that does not have cyclic AMP bound, there's a little pocket where cyclic AMP can bind. And once cyclic AMP binds in the pocket of protein kinase A, it activates that enzyme kinase. And so kinases are responsible for phosphorylating proteins. So protein kinase A has a lot of different protein targets that now it can go and phosphorylate specific residues, and that will either turn on or turn off that protein. Completely depends on how the phosphorylated state relates to the activity of that protein. But just think that a bunch of different proteins shift from being in an off mode to an on mode, and some that were in an on mode turn off, which causes ultimately a cellular response. Again, this could be that the muscle relaxes or the cell relaxes. It could be that it exports some things into the bloodstream. Like that could be the ultimate cellular response. So again, kind of just breaking down the different steps from the overview. The receptor is a G protein coupled receptor. The transducer for the signal is the G complex. In this case, it's the alpha subunit of the G complex. You'll see that in tasting. It actually, the beta and gamma complex, once they're released, they go and bind the effector. So it could be some of those, but in cyclic AMP formation, it's the alpha complex. So that's our transducer. The effector enzyme, so what does the transducer move the signal to and change, is the adenylyl cyclase. So adenylyl cyclase creates cyclic AMP being your secondary messenger, and then that cyclic AMP can go and bind to protein kinase A, and protein kinase A, its activity is turned on, and then therefore it can go around and phosphorylate a bunch of enzymes and change their state from active to inactive, or from an inactive state to an active state. Okay. Ooh, that was a lot. All right, so if we kind of look a little more at the transducer itself, so it's the G protein complex. It's made up of 3 subunits, alpha, beta, and gamma. And in the inactive form, in the alpha subunit, GDP is bound. So GDP, I have the 2 structures here. GDP is the diphosphate form, and GTP is the triphosphate form. So we have our 3 phosphates or we have our 2 phosphates, right? So GDP being bound, it is in the inactive form. Once the receptor makes that conformational shift, it'll change how these subunits all bind to each other and essentially releases. So notice that this is kind of more oval, and then it looks a little more triangular, kind of arrowhead-like over here. But so when it's released from the beta and gamma complex, it changes its shape, which allows GTP to bind. And then that activity, the transduces and goes and binds the adenylyl cyclase and turns it on. And eventually the signal is going to have to be turned off, right? You're not always going to have the signal in an on mode. You have to turn the lights off at some point. So in order to deactivate this alpha subunit, you'll have GTPase, which is an enzyme that cuts right here, this GTP, and makes it back to GDP, which turns off that transducer. So it removes itself, or it unbinds from the adenylyl cyclase, which then adenylyl cyclase activity is muted. And this in the inactive form can go back and bind the beta and gamma subunits, and then move back and sit on the receptor again and wait for the next signal to come. So that's about the chemistry or the biochemistry of the transducer. Here is the chemistry of the adenylyl cyclase. So adenylyl cyclase, again, converts ATP that's in the cytoplasm and then forms cyclic AMP and releases inorganic pyrophosphate, which is PPI. That's just these 2 diphosphates right here. All right, so we're going to form the cyclic AMP. So what is the mechanism behind that? And I'm telling this mechanism because you have to make a stop motion. Your first stop motion assignment is to do the mechanism of the formation of cyclic AMP. Okay, so again, cyclic AMP is our secondary messenger, and it's going to go activate protein kinase A. But if we look at the adenylyl cyclase active site, we can see, so this molecule right here is our ATP. So this ADE is the adenine ring. So that's that purine ring. Here's the ribose of our ATP molecule. There's the gamma phosphate, and then it says PPI right here. So there was the beta and the alpha phosphate. So they just shorthanded that part of the molecule, the structure, a lot. But so this is our ATP. And you can just see some interaction. So we have an arginine right here. It's arginine 1011 that is forming an electrostatic interaction with the closest phosphate. That's the one that's going to eventually come around and cyclize. And then we have an aspartic acid and asparagine kind of coordinating that alcohol that we're going to be also playing some chemistry on. Okay, but so mechanistically, what happens arrow pushing wise is the aspartic acid deprotonates, pulls that proton off of the hydroxyl group here and creates this oxyanion. And you can see now that the aspartic acid now has that H on it, right? So there's our proton, but we've created a really good nucleophile because anything with a localized negative charge on it is a really good nucleophile. So this nucleophile, our O minus, our oxyanion, will attack the gamma phosphate. Electrons will jump up on the double bond of the phosphate, jump down, and then kick off some leaving group. And the leaving group they're going to kick off is that inorganic pyrophosphate, that PPI. This PPI, though, on the way out the door, and actually it'll grab that proton from the aspartic acid to regenerate the active site residue so that it's back in its negative charge. And also that just corresponds to the correct protonated state of diphosphate when in the cytoplasm at physiological pH. Okay, but again, so just quick recap: aspartic acid grabs proton, creates an oxyanion, really good nucleophile. This attacks this gamma phosphate. Electrons jump up on the double bond, jump down back to the phosphate, and have to kick something off. And they're going to kick off inorganic pyrophosphate, that PPI, or the diphosphate. And that's how you create this bond right here, right? That was my oxyanion, and there's the phosphate that is now attached to, and then there was the leaving group. So this is the final cyclic AMP structure. So that is the arrow pushing mechanism of adenylyl cyclase. Okay, so the secondary messenger, cyclic AMP, again, goes to bind protein kinase A. And then what does protein kinase A do? Well, protein kinase A plays a role in activating or turning off all different types of enzymes and lots of different types of pathways. So it plays a direct role in glycogen synthesis, in glucose and palmitate synthesis. Let's see, it also can phosphorylate upstream kinases that go and play a role in something else. Again, that signal transduction is kind of like a little spider web of points. Mitochondrial functions can be altered. This plays a role in the inflammatory response, so it's going to turn on a lot of different things. Again, it just totally depends on what cell type you're looking at. Okay, and the last big thing to take away from signal transduction, which is sometimes like a multiple choice question, is the whole point of signal transduction is signal amplification. So what does that mean? It means that one tiny initial signal, one hormone molecule can bind one receptor. So there's our G protein coupled receptor. And then the G complex can go and activate multiple adenylyl cyclases. Okay. And then those multiple adenylyl cyclases could create multiple cyclic AMPs. So notice we've gone from one tiny hormone to now 1, 2, 3, 4, 5, 6 cyclic AMPs. And in fact, it's way more, but it's again, you're amplifying the signal. You're making it bigger and bigger and bigger. And then that cyclic AMP can go and bind this protein kinase A, and that protein kinase A could go and activate another protein kinase A, downstream kinases. And so again, you're just amplifying the signals all over. So signal amplification is something you need to keep in mind when you're talking about signal transduction. It's typically kind of like a buzzword that likes to get thrown around, like I said, in some multiple choice questions. So keep in mind signal amplification. Taking one hormone and then creating lots of secondary messengers that go and affect lots of different things. Okay, so how do we stop the process? How do we dampen the signal? And there were kind of two things that have to happen to like dampen this signal, turn it down. Okay, we're done. First thing is eventually this ligand will be removed from the membrane receptor. It'll eventually unbind and then go away, which will create another conformational change to allow the G protein complex to rebind. Remember, there's a GTPase that'll cleave GTP and form GDP, so then that can release from the adenylyl cyclase and go away. But the other thing is how we like dampen the signal and like stop this whole process is the use of phosphodiesterases. So phosphodiesterases are going to cleave the ring of the cyclic AMP to give you 5' AMP. So 5' AMP the 5' means that the phosphate, the one single phosphate's on the 5' carbon of the ribose ring. So 5' AMP cannot activate protein kinase A, right? So then that's going to stop protein kinase A's activity, and that's going to dampen and stop the signal. So that's how we can dampen this whole signal transduction pathway. Okay. The second signal transduction pathway that I want to look at that is coupled with a G protein coupled receptor is the IP3 DAG pathway. IP3 stands for inositol triphosphate, and DAG stands for diacylglycerol. And this structure right here is the phospholipid that's in the inner leaflet that's going to give us the inositol, which is a sugar, triphosphate, 1, 2, 3, and our diacyl, 1 acyl, 2 acyl, glycerol. Here's that glycerol molecule here. Okay. All right. So in this pathway, a lot of very similar things occur. Actually, let me go ahead and put the like breakdown of the different parts. Okay, so we still have a G protein coupled receptor in the membrane, integral membrane, and the ligand will come and bind, whatever that is, a hormone or a small molecule will come and bind, and that will create a conformational change, which will release the G protein complex. When the alpha subunits release, GDP goes away, and then GTP binds, and that gives the active form of the alpha complex. This then binds PLC, which stands for PLC cuts the that molecule, the phospholipid I just showed you. Phospholipid. Oh man, I'm so sorry. It is escaping me right now. phospholipase C. I think that's it. Phospholipase. It's a phospholipase is like an enzyme that would cleave a phospholipid, and then C, I just think is the letter of like, that's the third isoform of it. So it's C, phospholipase C. So phospholipase C is an integral membrane protein which finds that N-acetyl fatty acid, and it'll cleave that phosphate off. Boom. Or the sugar moiety with the 3 phosphates off. So you get your IP3 secondary messenger, but you also get the diacylglycerol secondary messenger, right? Without the sugar and the phosphates on it. So you have your diacylglycerol, that's DAG right here. Okay, so that creates 2 secondary messengers, IP3 and DAG, because they both do different things. So let's follow DAG. So DAG stays in the membrane because it's part of that phospholipid. It's just missing a sugar in the phosphates now. But so we have that diacylglycerol, and that binds protein kinase C. So protein kinase C would be your effector enzyme. And again, it's a kinase, so it's going to phosphorylate proteins, and that's going to switch them from an on state to an off state, or from an off state to an on state. So it's going to change the activity of different proteins. So protein kinase C is different from protein kinase A, which is in the other one, right? It's just a different isoform. It's going to have different targets. Okay, so that's diacylglycerol goes and turns on protein kinase C, and IP3, that's the sugar with the 3 phosphates on it, is going to travel through the cytoplasm over to the endoplasmic reticulum and bind to calcium channels. And that is going to release calcium from the lumen of the endoplasmic reticulum into the cytoplasm. So that can do a few things. You're going to increase the positive charge all of a sudden within the cytoplasm, and that can actually depolarize the cell because you're increasing the positive charge inside the cell. So you can have like a depolarization effect, which is what you'll see in the tasting. When you taste things, that's what happens. That's how you create an action potential, is by depolarizing the membrane there from IP3 binding and calcium flooding into the cytoplasmic region. Other things that can happen, right? So if you increase the calcium, it looks like calcium is also going to bind our protein kinase C to be active. So that'll also help activate protein kinase C in a way. And so that's like the big kind of 2 takeaway things. So our receptor was another G protein coupled receptor, and that's because a G protein is coupled to the receptor. The transducer of the signal is the G protein. This is the alpha subunit particularly. The effector enzyme is the phospholipase C, which is going to use this phospholipid right here as its substrate and cleave off the sugar and 3 phosphate moieties to give you your secondary messengers. There's 2 and eventually 3, but the 2 immediate secondary messengers are IP3 and then DAG, so diacylglycerol, and then the N-acetyl triphosphate. IP3 binds calcium channels and opens them up in the endoplasmic reticulum, and calcium floods into the cytoplasm, which can elicit some type of cellular response. And it could be something like creating an action potential would be the cellular response. And then diacylglycerol binds protein kinase C, and protein kinase C goes around and changes the phosphorylated states of proteins, which switches their activity, again, from an on to an off state or an off to an on state, which will cause some type of other additional cellular response. Calcium in this example is also acting as a secondary messenger because it's going to take the signal and make an ultimate cellular response. So in some multiple choice questions, the cellular response is not increasing calcium in the cytoplasm. It'll be calcium's acting as a secondary messenger in a sense. Again, the ultimate cell response might be something like cell depolarization, or calcium activates this other enzyme that needed calcium, kind of like this protein kinase C that needs calcium to work and function. Okay, so kind of breaking apart a couple pieces here, a little quicker. Again, it's the G protein complex, so it's in inactive form. Once the signal is received and the receptor has a conformational change, it allows these 2 to dissociate from one another and GTP to bind, and then the alpha sub complex becomes active and then goes and binds to phospholipase C. Here it is. Okay, so phospholipase C, that's what it was called. Here is the substrate. This is the phospholipid. Again, it is the phosphatidyl inositol. So here's the inositol, which is a sugar moiety. It has 3 phosphates, 1, 2, and there's the 3, which is part of the phospholipid. Okay. And then R1 and R2 are our long acyl chains over here. Oops. All right, so here is our substrate for this enzyme. And again, it's going to take a water molecule and cleave and put a water molecule. Actually, water will attack this phosphate. Electrons jump up, jump down, and kick off this oxygen. And so you see that part of the water molecule, right, is that oxygen is now part of the phosphate of the IP3. So the sugar with the 3 phosphates, 1, 2, and 3. And then the other one of the hydrogens is this hydrogen right here on our glycerol, 3 carbon chain, each that eventually at one point had alcohols, but when you attach fatty acids to it, those alcohols become ester bonds. So here's the diacyl part, and then glycerol. So those 2 are our secondary messengers. Remember, diacylglycerol activates protein kinase C, and the IP3 activates the endoplasmic reticulum calcium channels and opens them up. Okay, so how do we dampen this signal? Well, one thing we can do to dampen the signal is if we remove IP3 and via phosphatases. So phosphatases are going to come in and use a water molecule and hydrolyze off each of the 3 phosphates. So IP3 turns into IP2 diphosphate, which turns into IP monophosphate, and eventually you have all 3 phosphates are removed and you're just left with that sugar molecule. So if you down the concentration of IP3, right, that's going to allow these to dissociate more, and eventually that calcium channel is going to shut. There will be an influx of calcium in the cytoplasm, but that will eventually be restored. It'll get sequestered again back into the endoplasmic reticulum or wherever else it needs to go. And again, lowering the concentration of calcium in the cytoplasm will remove calcium here, and that'll eventually also deactivate protein kinase C because it needs calcium in order to work. Okay, so some takeaways, which I don't think I finished writing. So I'm going to post a link to this, and I would like for you to make sure you read over the different takeaways. The end.